Good day, sir.
Good day.
Thank you for being on Africa Researchers Magazine.
Anytime.
Can you please introduce yourself?
Okay, my name is Beckley IKHAJIAGBE. I am a lecturer with the University of Benin. I have a doctorate in ecophysiology and bioremediation technology. I'm a member of the Nigeria Bioinformatics and Genomics Network. I'm a fellow of the Linnean Society of London. I'm also a fellow of the Institute of Policy Development and Management. I am an executive member with the Edo State branch of the Nigeria Environmental Society, Botanical Society of Nigeria, Genetic Society of Nigeria, and the Biotechnology Society of Nigeria. The editor-in-chief of the Journal of Underutilized Legumes is the official journal of the Society for Underutilized Legumes.
You're welcome, sir, once again.
Sure.
Today we'll be discussing with you on the topic environmental restoration and food security, the role of bioremediation. Sir, the world today is bedeviled with the impact of global warming. This, as many experts say, emanated from the uncontrolled industrial processes and indiscriminate waste disposal, leading to worldwide pollution of the air, land, and sea. Do you agree with this assertion?
Yeah, 100%. I do.
Can you throw more light, sir?
Okay. The thing is, nature is superb. Nature is good, but sometimes one begins to wonder whether man and the environment are actually a perfect mix, because wherever man goes, unfortunately, it's well, if it is not too bad, I could say bad news, because we always want to conquer the environment. You want to build houses, you want to change nature just to suit your own economic purpose, and at the end of the day, always come up with habitat degradation. And today we are faced with huge problems of ecosystem loss, and it's so terrible now that our carbon sinks are almost going away, and man usually would want to conquer the environment. He always wants to make himself felt. Not for any reason, but perhaps to satisfy one economic need, one aesthetic need or the other, he ends up destroying the environment, remodifying the environment, not necessarily knowing that his actions may lead to habitat loss. And currently, our carbon sinks are gradually disappearing, and that has led to increase in global temperature, and that is actually beginning to influence climatic change patterns. And whether you like it or not, that eventually is a huge blow on international food security.
Okay, sir. So far from your explanation, I can deduce that the earth has a problem, and the problem is man, we humans. And
yeah, let me quickly interject.
Okay.
Well, not really that we are the problem. I think that because we are the most advanced species, if we take it right, if we do it right, then fine, things will be okay. It's not really that there is no possibility for changing natural environments, because as much as something new comes into an environment, there's always that possibility for one change or the other. But the thing is, we ensure that it is not so much that it begins to affect biodiversity, status of the environment, and eventually leading to huge or humongous negative impact on the earth itself.
Okay. So if we are to go by what you're saying now, it's possible that there can be deliberate acts of recovery from human perspective. Yeah. Is this achievable?
Of course. First and foremost, we identify that there is a problem, and we already know that the reason for that problem is what we caused ourselves, whether we like it or not. We came up, took over specific environments, conquered those environments, modified those environments, and then all of a sudden we had significant biodiversity loss, we had significant changes in ecosystem processes and all that. And what else did we expect? It's terrible, but we can retrace our steps back and do the right thing. And I think that is where the aspect of remediation comes in. Let me quickly put in that June 5 is World Environment Day, and the theme is reimagine, restore, recreate. Those three words are synonymous with getting back what we had destroyed, putting back the earth taking the earth back to where it was and what it really should be if we really want the best out of it.
All right, thank you, sir. I've taken time to go through some of your publications prior to this interview, and I've seen some of your publications on ResearchGate and Google Scholar. I discovered that majority of your work is centered on environmental restoration, and particularly with the use of plants and microorganisms. I'm sure this process is what is called bioremediation.
Yes, bioremediation, the process where you use biological entities to achieve any significant cleanup in the environment. Bioremediation. You could also remediate using other means, maybe physical factors of the environment to remediate, changes in temperature, modifying pH in the environment, processes as simple as mopping or even tilling the earth, those physical processes, or direct chemical interventions, adding surfactants to the environment, just to reduce surface tension of contaminants, particularly organic contaminants. All those processes would usually help to reduce or perhaps to remove contaminant presence in an environment. But usually, as proponents of nature, we always insist that remediation processes must be biologically based, because we are sure that it is only with such process that you can be rest assured that things are done properly. Because with bioremediation, the fear that contaminants that you try to remove will not be entirely removed from the environment. Like, okay, let me just take one example. If I was to use, say, I had oil in water, I probably want to boom so that I foster spread of the oil. And then I want to use a skimmer to skim up the oil. But you will not be too sure that some of those water-soluble fractions of the oil would have gone into the water. So, but if I used bioremediation processes, say I used probably microorganisms, plants, or even interactions between microorganisms and plants to remediate, then even the ones that are water-soluble would have been taken up by the plants, they would have been metabolized, and all that. So we actually would want to go for bioremediation.
Okay. So, with the current pollution problem we have in Nigeria and the world at large, do you think bioremediation might just be the best solution to the problem?
Splendid. I 100% go for it. The thing is, I say it is not as if you lay hold on the other forms of remediation, you couldn't achieve all you wanted to. But even in the scientific world, we say that one of the reasons why we want to take bioremediation over physical forms of remediation or chemical forms of remediation are because the process is entirely ecosystem friendly. You want a process that will not, add more trouble to the environment while you are trying to clean it up. Like the saying, rubbing Peter to paper. With bioremediation, that's not rubbing Peter to paper. That's actually doing the job.
Okay. Thank you, sir. You mentioned in one of your research that the capacity of plants to clean up polluted soil and also survive even in unfavorable soil conditions depend on the quality of rhizospheric microorganisms associated with the plants. Why is this so?
Let me start with this very funny term. They said behind every successful man is a woman. Yeah. So we would also say behind every successful plant in remediation are microorganisms.
Yes.
So it's almost as if the microorganisms are the wives of the plants. But actually what happens is every plant or even the microorganisms usually associate with other organisms. Usually we call them association. There are different levels of association. But in this particular case, we are looking at symbiotic associations where both the plant and the organisms benefit in the association. So imagine that you have organic contaminants in the soil and then the plants are not able to metabolize those contaminants, implying that the plants eventually will die or would have impaired forms of metabolism, which usually will not be too good for the plants. What the plant usually does is that it gains from its association with its rhizospheric contents, like the presence of plant group promoting rhizobionts, like Pseudomonas. Most Pseudomonas are good examples of plant growth promoting rhizobionts or rhizobacteria. What they do is that they enhance plants' capacity to exude compounds that will remediate whether organic contaminants or not. There are cases where even the plants may not be able to remediate the contaminants, but the coexisting bacteria or fungi in the root system of the plant are able to exude contaminants that will loosen up the substances. A good example is in the release of biosurfactants like rhamnolipids, lipoproteins, and other compounds that will help to loosen up surface tension, and then thereby enhancing capacity for the plants to metabolize those organic compounds. Well, also suffice it to say that the bacteria would also make good use of the carbon contents of the organic contaminants. Well, let's leave organic contaminants. And for inorganic contaminants, we know that plants will take up anything that becomes ionic, because if any compound becomes ionic, probably because it has been dissipated in its ions in the presence of water, the plant is able to take it up. But the question is, how do you differentiate between a remediator plant or a sensitive plant or a plant that just takes up the inorganic contaminant and dies? Yeah, because I said all plants have capacity for taking up contaminants, minerals, micronutrients, macronutrients. The question is, does it survive after accumulation? If it doesn't survive, then obviously you don't want to use such a plant. You always want to use plants that will survive. Now, it is not as if all plants have the same capacity to take up contaminants, no. But because of the presence of their rhizospheric organisms, some of those plant growth promoting rhizobacteria or fungi would help to release what we call chelates or chelatins, biochelatins, that we bind with those inorganic contaminants and thereby enhance absorbability of those contaminants by the plants. And then the plant eventually takes them up at much more faster rates, and all that. So that marriage between plants and the rhizospheric microorganisms are something you really don't want to ignore, particularly when you do bioremediation.
Okay. Thank you for that elucidative explanation.
Sure.
In one of your papers that you recently published, titled The Growth of Rice in Vitro After Inoculation with Bacterial Isolates from Typica ferruginous utisol, was published in the Bulletin of National Research Center, you reported enhanced rice development with rhizole inoculation using plant growth promoting bacteria. You also have about 10 articles hereabouts that you published between 2020 and 2021, where you reported similar enhancement of crop growth under rhizole inoculation regimes. Can you please share light on this drive as a means of achieving food security in a polluted soil?
Yeah, most farmers are so key. They are so this drive for acquiring NPK fertilizer. Probably you need the nitrogen to enhance foliar development, vegetative development. But when it comes to yield of the plants, yield components, yield factors, you want to enhance potassium and phosphorus accumulation. Now, unfortunately, is the fact that though the soil may be rich in any kind of soil, may be rich in phosphorus or phosphates, which are actually necessary for better yield components, those phosphates are never biologically available for the plants. So usually most of the plants would rely on plant growth promoting rhizobacteria that have the capacity for even free living bacteria, not only rhizospheric, even free living bacteria. But insofar as they have the capacity for phosphate solubilization, they help solubilize the phosphates and then the plants take it up. Now, in most of the studies I did with ferruginous soils, by the way, by ferruginous soils we mean soils that are high in iron. We observe that once the soil has too much of iron, it usually will affect the metabolism of phosphates or the release of phosphates in such soils for plant use. So what we did was to use microorganisms that have capacity for survival in those highly ferruginous soils, particularly the ones that produce lots of components that would enable their metabolism in such soils. And then the organisms would also have capacity for high phosphate solubilization. So we rhizo-inoculated them, and we observed that their yield components were more than between 25% to 75% higher than plants that were not inoculated with such bacteria. Some of them, I cannot be able to tell the particular strain, but we had up to 2 strains, one from Pseudomonas aeruginosa, then another from a specific species of Bacillus. Then what we have also succeeded in doing is, for that of the Pseudomonas aeruginosa, we were able to develop microbial inoculants from it, so that in cases where it can be made available if say peradventure somebody has to plant say rice in ferruginous soils. All you just need to do is rhizo-inject the inoculant in the soil. Phosphate solubilization becomes easy, and then yield component becomes enhanced. Well, again, it's still at the pilot research stage. By the time we are done with it and we do lots of trials, we should be able to come out with something better for farmers.
Okay, that would be nice, and we'll be looking forward to that.
Sure.
Prof, as a bioremediation enthusiast myself, I've always hoped that the economic values of crop plants could be enhanced when they also double as a phytoremediation of pollutants. That way, we'll be using one stone to kill 2 birds. What do you think, sir?
It's easier said than done. It's actually using one stone to kill 2 birds, but I can assure you are using one stone to kill 3 birds. You are killing 2 birds, and you are killing the human being that is throwing the stone.
Can you expatiate on that, sir?
So it's the thing is this. As I said earlier, I said all plants have capacity for taking up compounds. They can take up inorganic compounds. Insofar as those compounds have been dissipated in their ionic forms, the plants will take it up. But the question is, will the plant die? Some plants don't die, and so for that reason we say such plants are either tolerant plants or highly resistive plants. Otherwise, if they die, then they are sensitive plants. And I'm very sure that nobody would want to take up plants that would die, because obviously the plants would have even gone off before it were time for harvest. So peradventure you have a plant that is resistive or resistant to metal accumulation, and so such plant is able to be used for, the plant is used for remediation purposes. There's a huge problem. The problem is that you really don't know where the plant dumps those components, those compounds. I know that in some cases, some plants biotransform the compounds and turn them to maybe highly organic forms of inorganic contaminants. If I give an example, some plants are able to take up cadmium. Cadmium usually would not be necessary for metabolism, so they'll be dangerous and can instigate cancer. But what some plants do is that they complexate cadmium so that inside that plant, cadmium does not cause any metabolic harm. But what about humans? Because you really don't know whether the kind of degradative enzymes in the GI of humans or even animals may have capacity for dislodging whatever complex forms you find those metals. So the problem is, we cannot be too sure. Most of those metals will find themselves in harvestable plant parts that eventually you would use, you eat, and then at the end of the day you have bio concentration of specific metals in the food chain. Before you say Jack Robinson, you have actually killed 3.
So, Prof, please, I'm trying to wrap my head over this. In a layman's language, are you trying to say the possibility of using some plants, let's say food crops, for phytoremediation might lead to the plants may absorb this contaminant and pass it on to man when consumed?
Very sure. It is not all the time. It's not all the time, because the truth is, if I know as a researcher, I know some plants that would biotransform certain contaminants. I also know some plants that will accumulate contaminants only in the leaves. Some will accumulate contaminants only in the roots. Those cases, I'm sure the term rhizoextraction, phytoaccumulation, comes up usually when if I was allowed to talk more on bioremediation. So if a plant is highly known to rhizoextract and then keep those contaminants within the rhizospheric region, then such plants to a large extent, well, not to a large, to some extent, might be safe, because that means that the plant would have excluded the contaminants from the leaf. But then the question is, how do you know which plant does that? You will not be too sure, because this capacity for exclusion, excluding metals from leaves, excluding metals from roots, excluding metals from certain harvestable parts, vary from plant to plant, even within plant populations. We have seen situations where environmental factors usually would affect the capacity of such plants. Well, because sometimes such characteristics are genetic, and you and I know that the expression of certain genetic characteristics are environment-based. The environment may favor expression of certain capacities, and you'll not be too sure. A particular plant does a particular thing in one environment, but doesn't. A good example is with, I think a very fast example is with African yam bean. You could take one specific type of African yam bean, the same genome, the same gene, you plant it in Benin City, it will tuber. If you take that same plant, you take to Ibadan, it will not tuber. So what I'm saying is, changes in environment will affect capacities for gene expression, and so nobody wants to take that risk. You don't want to be you don't want to be caught up in the middle. So it's just better, you want to do remediation, use plants that will not go back into circulation in the food chain, so that you are sure any aspects of bioaccumulation of metals in the food chain is, knocked off. So we don't in any way support the use of any plants that you will find in the food chain, in any ecosystem whatsoever.
For phytoremediation?
For phytoremediation. No. Okay. Because you can't be too sure that you are not going to recycle the contaminants.
But, Prof, sorry to interject at this point. What we're trying to look at is have a scenario whereby we can have a plant that would also serve as food for man to, take care of the timid population in Africa and provide food security. And these plants should be able to grow in polluted soil, which you agree with me, we have a lot of polluted soil in Africa. Don't you think it's important at this point to begin to look into that research area of plants that could carry out phytoremediation without the contaminants getting to the fruits?
It is not as if it is not possible.
All right.
It is possible. But would we continue to die in hunger while we do that research? It's not as if it's not possible, it's possible. What the thing is this. In genetic engineering, that is absolutely possible. You could bioengineer a particular plant to probably restrict its accumulation to the roots. If we say vegetables, say the amaranths, pumpkin, and all that we commonly consume, if you bioengineer such plants and ensure that all contaminants are restricted to the roots of such plants, because you don't need the roots, you only need the leaves and perhaps the stem, then that's okay. But then you also need to think of something. You also have to be sure that there is already this hulabaloo about GMOs. If I come out with a bioengineered plant that has capacity for remediation, has a way of either transforming contaminants or excluding contaminants from harvestable plant parts of economic importance, and I tell the world that it's ready for consumption, you can be sure that some other persons will come and say, well, because it's GMO, nobody's going to take it. So that's usually one problem we're going to face. Now, it still does not mean that if you do definitive, purposeful, continuous breeding, selection, and all that, you may not be able to arrive at that. But you must understand this. Whatever you do has to be meta-specific. You must understand that we have more than, particularly in the periodic table, you have more than 30 metals that plants usually would not want to use, microelements, and also microelements of health significance for humans and even for animals. So we are not talking about one plant. So if I want to do this kind of research, then you must understand that it has to be meta specific. So if I have a plant that is able to seclude lead, What about when in that same soil you have higher concentrations of zinc? What's going to happen? So just think that the first thing is that we must get the things right. Ensure the environment is clean first. You want to plant, you want to get crops available just to ensure that we achieve food security. Then we do the right thing. First, reduce pollution. Reduce pollution. And if you want to remediate, you remediate with plants that are not going to be found back in the food chain. I think that is really sacrosanct, because by the time we keep talking about, trying to use one stone to kill two birds, giving plants capacity for remediation, and at the same time you use the plant, you eat the plants, again, you cannot be too sure whether digestive juices or enzymes in the GI guts of the herbivore or even the omnivore might not just be enough to set back, that cycle of contamination, releasing the so-called sequestrated contaminants in the plant back into the environment. I think, it's good topic for research, but for now, no.
All right. Okay, Prof. That is well understood. Do you think the government has a role to play in research regarding bioremediation, and how?
Yeah, there are a lot of agencies the federal government has set up that look at the possibility for cleaner environments. Some agencies and a lot of researches too that have been done, to enhance environmental health. But my problem is most of the researches we have, with all due respect, are either done for the sake of publications, or you find them on the bookshelves of most universities, polytechnics, and research institutes. I think government can take a step further. Government can invest in some of this beautiful research by researchers, plug it back into the system. I know that in most advanced countries, what they do is that they get some of the agencies and companies involved in research, so that whatever the researchers do, they get them plugged into their own industrial processes and protocols and procedures. I think if we do that, then it will be better. Researchers will be more encouraged to go deeper. Imagine a situation where in this climb, you hardly get enough grants for study, and then by the time you do the little you can, you want to publish, it's that expensive, and I know the government can do better. It's not as if government is not trying. I know that we've had situations where there have been a couple of grants from TED Fund and other grants bodies, but the major problem is, even after getting that from TED Fund and we are done with the work, it's either published or left in the shelf. We don't see them. Researches are not commercialized, so government can take a leap from that. Commercialize research. That's just it. Not leaving them on the internet or even on the shelf. Commercialize research, and then we are done.
Thank you, sir. I want to appreciate your time on African Researchers Magazine. You have shared your wealth of experience with us, and we do really appreciate it. We want to appeal at this point that we can call on you some other time to help us elucidate another scientific research topic. So thank you again for being on African Researchers Magazine.
Thank you.
Thank you, sir.
Sure.